Welcome to the Da Vinci Academy Histology video course. The entire video course is available on YouTube and covers all of the fundamental principles of histology and relevant cell biology. You can find all of the videos from the course by clicking the Histology playlist link in the description below, and then you can access the corresponding practice questions and histology lab videos by going to our website, which is also linked in the description below. Alright, so this is the second of the blood lectures, and in this lecture we're going to cover the white blood cells. So white blood cells, or WBCs, they are key players for the immune system. They're also produced in the bone marrow like erythrocytes, and they can really be distinguished into a few categories. So you got lymphocytes, which we'll talk about first, and then you've got neutrophils, you got eosinophils, basophils, and then you've got monocytes / macrophage. And we'll talk about all these. Monocytes, the inactive form, macrophage is the active form. So lymphocytes, they consist of three cell populations: T cells, B cells, and natural killer cells, or NK cells. And histologically, you can't tell a T cell from a B cell from an NK cell, especially in a peripheral smear. In connective tissue, you can see an activated B cell, also known as a plasma cell. We'll talk about that again in a little bit here. We actually talked about that in the connective tissue lecture. So cytology-wise, again, we talked about this previously, but just to review, you have a round-shaped cell like this, and you can see this is a great image of it. You have a dark, round nucleus with a condensed chromatin. So the reason it's dark is because you have that very condensed, tightly packed chromatin like this. So dark purple basophilic nucleus like this, and that's due to the heterochromatin. And then here, this is a great image of it. You can see this kind of thin rim of paler cytoplasm around the nucleus like this. And you can see here's another image of it like this. And again, if you kind of use the red blood cell as reference, you can see it's a little bit larger than a red blood cell normally. So T lymphocytes, like we mentioned, they're produced in the bone marrow, but they mature in the thymus. So T for T. And then they comprise 25 to 35% of white blood cells. And then they're broken into two major categories: CD4 positive T cells, which are also known as T helper cells, and then CD8 positive cells, also known as cytotoxic T cells. And there's a lot of details here as far as immunology goes. We're going to walk the very fine line of kind of giving you the basics, the very bare minimum basics that you'll need to understand the differences between these two. But just know that there's far more detail involved that we'll leave to the immunology courses to teach you. So for CD4 positive cells, we've got a nice diagram here to show you this. What they do is they recognize and bind foreign antigens that are bound by major histocompatibility complex, also known as MHC2. So there's MHC1 and MHC2. So CD4 positive cells recognize MHC2, and these are present on the surface. These are essentially proteins that are on the surface, as you can see here. They're on the surface of antigen-presenting cells. So basically, antigen-presenting cells are cells that engulf foreign material or foreign agents like microbes, viruses, and then will break them down and present components or antigens, foreign antigens, to T cells. So here you have an antigen-presenting cell. An example of an antigen-presenting cell would be dendritic cells. Macrophages are major antigen-presenting cells that go around eating up foreign material and then presenting it to T cells. And then B cells can also be antigen-presenting cells. So you have an antigen-presenting cell like this. You have a microbe. Let's say it's some type of bacteria. It gets engulfed by the antigen-presenting cell. Then within there, it gets broken down into smaller components, which would be the foreign antigen. And then the foreign antigen binds to the MHC2, which then displays it on the surface. So you can see here's the antigen-presenting cell essentially presenting the antigen, it's in the name, to the T cell. So you have the CD4 positive or T helper cell here. It has a T cell receptor that will bind and recognize this foreign antigen. And then beyond that is much more detail than we want to get into, but it helps coordinate an immune reaction to this cell and to this foreign agent or this microbe or bacteria. Now, as far as CD8 positive T cells, they recognize MHC1 on all nucleated cells that present self-antigens. They're essentially going around and checking every normal cell in the body to make sure it's not infected, to make sure it hasn't turned into cancer, to make sure there's not something globally abnormally wrong with the cell. So you can see here, here's an MHC1 that's presenting what's called a self-antigen. So this is a component from a protein that is a normal protein within the cell, and it's essentially signaling to the CD8 positive cell that, hey, the cell's okay, you don't need to intervene. Now, what can happen though is that these normal cells can get infected by a virus per se. So the virus comes in, it gets broken down into foreign antigens here, which then get displayed by the MHC1, as you can see here. And so this foreign antigen gets presented to the T cell receptor on the CD8 positive T cell, or the cytotoxic cell. And again, the exact mechanisms are beyond the scope of this course, but what you want to know is that this actually just very directly will lead to destruction of this cell. This can also be a way to defend against cancer cells. So if cells transform into cancer cells, the same thing can happen. You can have antigens from abnormal proteins or mutated proteins be presented, and then the CD8 cells can recognize that and destroy the cancer cell. And so again, as we say that here, the goal is to destroy abnormal cells within the body, such as virally infected cells, cancer cells. So B lymphocytes, these are produced in the bone marrow and they mature in the bone marrow. That's the big difference between T cells and B cells. So B for bone marrow, and then remember T for thymus. Function, they activate to become antibody-producing plasma cells that can also act as antigen-presenting cells, as we talked about. Key thing to remember here is that a B cell does not become a plasma cell within the circulation. It is after they have entered the connective tissue and then become activated, such as by a CD4 positive, a T helper cell. Plasma cells, remember we talked about this earlier, but they have the following appearance on histology. They have an eccentric nucleus. Remember I described it as almost like a ghost. So you have this nucleus out here and then kind of this apron like this, and you can see that here in this section right here. Notice both of these are from connective tissue. They're not from peripheral blood smears. And then the nucleus, it's eccentric and it has clumped heterochromatin, or a clock face nucleus. You can see that really well here. You can see it's almost like you have the numbers outside here on a clock face. And again, see how you kind of see it, the cytoplasm fanning out like this. What's nice in this section is this yellow arrow is pointing to this. See this kind of fainter staining portion right here? This is pointing to the perinuclear clearing, which is actually where the Golgi would be located. So that's just a nice bonus finding here on this section. Natural killer cells, there's not a whole lot you need to know about this. Again, you're not going to be able to distinguish them on histology. Just know that they are cytotoxic immune cells, so they're similar to CD8 positive T cells. They destroy normal cells that were transformed. So again, like, cells that were infected by viruses or have cancer. And they do this by secreting porphyrins to disrupt the cell's membrane. So what happens is if you have, your transformed cell like this, and then you have your NK cell like this, and so it's going to secrete these porphyrins, which are essentially going to poke holes in the membrane and allow water to very easily come into the cell and cause it to burst and destroy the cell. So next we'll talk about neutrophils, and actually the majority of white blood cells are neutrophils, about 55 to even up to 70%. They're the first white blood cells to fight infection and inflammation, so they're essentially the first responders. That can be a test question, so definitely keep that in mind. They work by engulfing microorganisms and necrotic tissue. Obviously, this corresponds to fighting infection, and then if you have tissue damage or tissue necrosis, obviously that's going to trigger inflammation. And so again, they'll work by helping to kind of be part of the cleanup crew in a sense of clearing out that necrotic tissue. They destroy engulfed material via lysosomal vacuoles. So they'll engulf material via phagocytosis, and then you have lysosomal enzymes, we'll say LE, in these vacuoles here, and the engulfed material will be merged with the vacuole containing these lysosomal enzymes, and that'll help break down this material. One thing to keep in mind is dead neutrophil and debris make up pus. So if you have a laceration on the skin and you notice that it has a bunch of pus, that's actually these dead neutrophils with their engulfed debris. And then just something to keep in mind here is phagocytosis by neutrophils can actually be enhanced when an antigen is coated in immunoglobulins that bind to neutrophil Fc receptor. So a process you're definitely going to want to be aware of that could definitely come up on a test is diapedesis. And it's the process where neutrophils in the circulation, so they have to get from the circulation to the tissue with an ongoing inflammatory response. So they have to be attracted to that situation and then also cross over from the circulation to the tissues with the ongoing inflammatory response. Here's a figure here that demonstrates the process. We'll go through each of these steps over the next few slides. So first, you can almost break these down into phases. You have the homing phase, where at the site of injury or inflammation, let's say it's a bacterial infection, you have these cells called macrophages. And we'll talk about these in a couple slides here. But macrophages, basically, they engulf foreign material, damaged material, and they obviously can present them to T cells, but then they can also secrete what's called cytokines, which are basically signaling molecules that are important for many processes in the body, including the immune system. And so what they'll secrete specifically is TNF alpha and then IL-1. And so these are the two major ones you want to be aware of. So they'll engulf, let's say it was a bacteria, and they'll break it down, and in response to that, they'll secrete these cytokines. What these cytokines do then is, so you'll have, your endothelium like this, which are just, an epithelial layer, and we'll draw, this is the other side, and you have the blood in here circulating. And so what these will do is they'll stimulate the expression of cellular adhesion molecules known as CAM, which would include E-selectin. So you can have E-selectin. E-selectin is almost like a roadblock, as we'll talk about here in a second. It kind of grabs the neutrophil and helps kind of slow it down. And then you also stimulate, these cytokines will stimulate endothelial integrins, which have a higher affinity. So these are more downstream. And so you have integrins as here as well that are expressed. So the E-selectins will kind of slow things down, and then as they're slowing down and coming to a stop, then they bind to integrins, which will bind much more tightly. So because you want to grab them as they're flying by, it's almost like if you think about a car on a highway. If a car on a highway is going really, really fast, you don't want to slam on the brakes. You want to slowly, like when you're about to get off at an exit, you slowly slow down, and then eventually, you'll get off the exit and stop at the stoplight. You don't want to just slam on the brakes. Same thing here. So you want to kind of slow it down and then eventually grab it at the integrin, and that's where it'll adhere to the red blood cell and then can begin the process of moving it to the tissue. So that next process mediated by these cellular adhesion molecules is called rolling. So they essentially slow it down, and then it's kind of in the name. It rolls across the cells because of its reversible binding. It hasn't tightly bound it yet. Then you have adhesion. So as it's rolling across, then it eventually will tightly bind to these integrins on the endothelium. And so you'll see that here where it binds, like right here. So these would be your integrins right here. Then at the same time, what you're going to have is perivascular mast cells, which will secrete heparin and histamine, which will help mediate this process. So, you have endothelial cells like this, and they're bound by, junctions. There's maybe a tiny little space between them. What you need to do to help this cell move from the blood vessel to the actual tissue where the inflammatory response is going on is you need to widen the space between these endothelial cells. So you widen the space like this, and then it allows the neutrophil to travel through these spaces and into the tissue. And so as a part of that process, then it enters into what's called crawling, or as we say here, transmigration. So they spread out via what's called pseudopodia, which is essentially these extensions or arm-like extensions that then allow them to pass through and kind of slide through these gaps like this between the endothelial cells. And then once they make their way in, then they're within the tissue and they can start engulfing foreign material. Again, we talked about this back in the connective tissue lecture, but just to review again. So appearance on histology, you have multi-segmented nucleus with pale pink cytoplasm. You can see that here. You can see multiple different segments. You can see in comparison, this is a peripheral blood smear. In comparison to the red blood cell, you can see the size difference here. Here's another one where here's a neutrophil here, obviously a bunch of red blood cells, and then here you have a lymphocyte here, so you can kind of see appearance-wise the different differences in appearance. Eosinophils, so these are the primary cells that are responsible for fighting parasitic infections, and then also along with mast cells and basophils, they help mediate allergic reactions as well. Appearance on histology, so they're going to have an eccentric bilobed nucleus, and remember we talked about this back again in the connective tissue lecture. It's kind of shaped like this, almost like a boomerang, and you can see that here. Here's a great example of it like this. So they have that bilobed nucleus, and then they have these multiple kind of red dots or eosinophilic dots, so bright red cytoplasm. You can see that here, and here's another great example of that here. So basophils, these are cells that contain high concentration of granules containing heparin sulfate and histamine, and this plays into their function, which is to mainly mediate hypersensitivity reactions. And they do this by binding to antigen IgE complexes via a receptor on their cell surface called FC. So the FC will bind to the antigen IgE complex. This will then induce the release of vasoactive substances, which would include heparin sulfate and histamine. And then this will trigger hypersensitivity reactions and anaphylaxis. So they're very similar to mast cells and often work with mast cells. And it makes sense. They're loaded with these granules containing these vasoactive substances. Once they get triggered, they're going to release those substances to carry out the hypersensitivity reaction. On histology, this is when you kind of have to see it a few times. So the nucleus can sometimes be hard to see because you have all these granules. But if you kind of look here, it's kind of a bilobed nucleus like that. And then it also will have what's very obvious is numerous dark purple granules. And these are these granules containing these substances. And so you can see here, you can see it's very granular, a lot of different granules all throughout. And then same thing here. So on a peripheral smear, again, histology is very visual. You got to just see some things a number of times. It's like radiology. You just have to see images a number of times to then be able to recognize them. And you got to see them in comparison to other cells or structures to help distinguish them. So here, this is a great example. You can compare a basophil to a lymphocyte to a neutrophil to a red blood cell. Obviously, it's full of red blood cells. So again, it gives you a sense of that appearance. So monocytes, these will circulate in the blood for several days, and then they migrate into tissues where they become macrophages, where they'll either reside and just kind of hang out and wait for something to happen, or if there's something ongoing, like an infection or an inflammatory response to tissue damage or something like that, then it'll fight the infection or take part in inflammation. The function, they do this by engulfing foreign material and tissue debris via phagocytosis, and then they act as antigen-presenting cells to present these molecules they engulf via MHC2 to CD4 positive T helper cells. And again, just to kind of illustrate that for you, let's say we have a bacteria here. So we'll say this is B, and then we have a much larger macrophage here, and let's say it comes in and engulfs that. So then here's the macrophage with the bacteria. It'll break it down into, smaller antigens. Those will get presented by MHC2, which then, remember, can be bound by CD4 positive T cells, and their T cell receptor, TCR, can then bind to that, and then it can trigger an inflammatory response to this antigen to help fight this bacteria. In fact, one of those responses, it can actually, through a series of signaling mechanisms that we won't go into detail about, actually trigger B cells or plasma cells to secrete antibodies to this specific antigen. Now, for appearance on histology, both monocytes and macrophages, it's going to be sort of similar. You're going to base the difference on location. Monocytes can be more so in the circulation. Macrophage could be in the circulation, but also definitely if it's in the connective tissue, you're going to call it a macrophage. And you're going to see a large indented nucleus, and you can see that here where it's slightly indented here. One way you can distinguish is macrophage. It's usually going to be a little more indented. It's going to be a little more kidney shaped, as you can see like that. And the nucleus is going to be kind of a large, irregularly shaped. It can sometimes be eccentric, as it is here. And then you're also going to have kind of a paler basophilic cytoplasm surrounding it. And again, just for comparison's sake, so here's a monocyte here, here's a lymphocyte here, here's numerous red blood cells surrounding it, just to give you that comparison there. Platelets, so this is the last type of cell we'll talk about. These are cells that aggregate to form a platelet plug. They're part of forming a clot. So let's draw this down here. So we'll draw a blood vessel here. So you have the endothelium like this down here. And what can happen is that you can have a break in the endothelium. So let's say this is a break right here. And below, remember, it's connective tissue. And so you're going to have what's, collagen. So you're going to have collagen below. Then you're also going to have what's called von Willebrand factor just below the endothelium. And so when you have a break in the blood vessels, you're going to have platelets get exposed to potentially either collagen or von Willebrand factor, and that's going to trigger platelets, which are kind of elongated shaped like this, to come in and aggregate in this region here. And so they'll aggregate in this region, and that'll form what's called the platelet plug, which is just an aggregation of platelets kind of sealing off. It's in the name, plugging off this break in the blood vessel here to prevent further bleeding. It's the initiation or the beginning of achieving hemostasis. And so the other thing that damaged blood vessels will do is they'll release into the blood clotting factors. And then clotting factors will stimulate prothrombin to become thrombin. And then thrombin will stimulate fibrinogen to become fibrin. So again, releases clotting factors. The clotting factors stimulate prothrombin to become thrombin, and then thrombin stimulates fibrinogen to become fibrin. And so over here, we'll mark this in black. Then you have these fibrin strands, which will come in and it helps seal off this platelet plug. So they'll form this meshwork and really firmly secure the platelet plug in place so that to achieve hemostasis and prevent any further bleeding. And then lastly here, just the appearance on histology. So these are anucleate. So like red blood cells, they don't have nuclei. They're flat, round cells. Sometimes they're elongated like I drew here, and they'll have dark purple spots. So you can see them here. Here's a great example. These are not nuclei. These are just cell fragments, and then they can also be granules that'll help mediate blood clotting. And so again, just the big thing is you don't want to get tripped up here and think these are nuclei. They're not. You can just tell that they're very small. You compare them to red blood cells, they're much smaller. And as you can see here, they're very pale with the exception of these dark blues, purple spots. Thank you for watching this video from the Da Vinci Academy Histology video course, which is completely available on YouTube. To access the corresponding practice questions and histology lab videos, go to our website using the link in the description below.